Welcome to the second part of our discussion about introduction to parasitology. In this video, what we're going to discuss is how the parasite cause infection to human and what is the mechanism, no, defense mechanism of our body against the foreign materials or against parasites. Now, remember guys that the function of our immune system is to protect the body from the invasion or from invasion by potential pathogens. Our immune system is a tight controlled balancing act in the sense that dysfunction of the immune system can lead to either permissive environment for infection on one hand or unchecked activation that can harm the organism on the other hand. Take note, okay, Take note that the immunity to parasites, especially eukaryotes such as helminths and protozoans, is complicated by the fact that unlike bacterial pathogens, okay, uh, what you call this, eukaryotic no, organisms are similar in the makeups and physiology. Okay, remember guys that parasitic infection in humans and animals occur when the parasite successfully establish itself to the host and is not eliminated by the host defense mechanism of an individual. Okay. Remember also, no, not all interaction between the host and parasites can result into injury. Okay. Not all interaction of parasites and to the host does not cause an injury. It it happen it can happen because of the following. Number one, the parasite fails to establish fails to become established to the host. Meaning to say, the immune system or the defense mechanism of the an individual is intact, no? Not to allow the parasite to cause an infection to human. Another reason on that is that because of parasite becomes established and the host eliminate the infection. There is an infection, no? There is the parasite, no? The parasite was able to uh, invade the human host or the host. However, the host itself was able to eliminate readily the infection. Another reason on that is because of the parasite becomes established and the host begins to overcome the infection but is not totally successful. Somehow, the human body or the host respond accordingly to the host or to the parasite. However, no, however, the parasite was still able to cause an infection, and the host was not able to totally eliminate the infection. Another reason on that is because the parasite established and the host is in trying to eliminate the organism becomes damaged itself. Okay. Remember people that the ability of the parasite to cause infection has evolved thoroughly. No? Through the process of natural selection, since only a portion of the parasites are able to accomplish this. Now take note, in the same way, the host itself naman has the ability to defend itself against a parasite invasion by uh, selection also. Remember, people, that some of the life cycle are complicated, that the parasite has adapted means to survive immune assault. Okay? Now, it is also important for us to remember that the host parasite relationship remains dynamic. That's why you cannot totally eliminate kasi dynamic siya. Pwede siyang magbago-bago. And while other parasites become specific to the host over the time, accidental infection happens. Okay? For example, the zoonosis. Example of zoonosis is trypanosomiasis. No? Trypanosomiasis is a zoonotic disease for animals, but now emerged as a parasitic infection to human. Another type is the newly discovered Plasmodium strain. No? And what is the Plasmodium strain? I'm sorry. Initially, but ayaw. No? Initially, we have the four clinically significant Plasmodium strains. We have the Plasmodium ovale, Plasmodium vivax, Plasmodium falciparum, and Plasmodium malariae. No? The dynamic of the parasite-host relationship was able to discover there are other parasites. And the fifth malarial species is the Plasmodium knowlesi. Now you know. Knowlesi. Okay? Now, remember that the natural physical barrier to the entry of the parasite in the body constitutes the first line of defense. And our first line of defense is, as you will discuss this in immunology, is no other than the skin. Helminths, no being a parasite, has the ability to penetrate the skin. And the skin penetration of the skin, I'm sorry, the skin penetration of the helminths allows it to invade the human body. For example of which is the strongyloides and hookworms. Strongyloides and hookworms synthesize the protein that aids their entry. No? Remember that the adaptive mechanism of some helminths allow them to overcome these defenses. Okay? Like the filariform of strongyloides and hookworm. Now remember that the cercaria of our schistosoma, no, are capable to also to penetrate the skin. The presence of glands in their anterior of the cercariae allows them to uh, secrete lactic enzymes, no? Allows them to secrete lactic enzymes that enables them to penetrate the skin. The mucus lines lining of the respiratory, of the gastrointestinal tract, of the urogenital tract, genitourinary tract provides barrier against parasitic infection. Okay? Tight conjunction between the epithelial cells serves as the preventive barrier to the passage of the smallest molecules. However, even though that we have there a protective barrier, no, parasites can also invade. For example, vagina, no, is known to be acidic in nature because of low pH of the vaginal secretion. And the acidic pH of the vagina enables the vaginal area to protect itself from bacteria and parasites. However, there are parasites that are resistant to low acidic level. Okay? Actually, Trichomonas vaginalis, no, is unable to survive in acidic vagina. So the question is, how come that this Trichomonas vaginalis is capable of causing infection, where in fact vagina is acidic in nature? The acidity of vagina is primarily because of the bacteria or the normal flora, Lactobacillus acidophilus. Okay? Lactobacillus acidophilus. This is the main agent, one of the normal flora of the vaginal area, which maintains the acidic level of the vaginal area. However, there are cases that this normal flora becomes insufficient in colony. Then that is an opportunity, no? That is an opportunity for my other microorganisms to invade and cause the vaginal acidity or the vaginal pH alkaline. Okay? That is why Trichomonas vaginalis is able to survive in the vaginal area because the pH becomes alkaline. On the other hand, we have the Giardia lambia. Actually, Giardia lambia is also affected by the gastric juices in the intestine. That's why Giardia lambia, from being trophocyte, has to become encysted. From trophozoite, it needs to be converted to cystic form. Why? So that this Giardia lambia will be able to survive. Because the Giardia lambia trophozoite can be diminished, the motility of Giardia lambia trophocyte can be diminished by the gastric juices. Okay? Therefore, reducing the motility or diminishing the motility of Giardia lambia can result to the reducing of injury that it can cause to the host. Okay? The ova of helminths are protected with acidic environment, are protected from acidic environment. That's why, di ba? You might wonder, you will ingest them, but they will pass through the gastrointestinal tract, knowing that the part of the gastrointestinal tract is the stomach, where the gastric juices are there, and acidic in nature can destroy yung food niya na may metabolize na eh. But remember, these helminths, ascaris, trichuris, tania, and others are protected against acidic environment. Okay? Why? What is with them? Because they are embryonated. Aside from that, they have thick shell covering. No? Cysts are resistant to acidic pH in connection to Giardia lambia. Okay? Si Giardia lambia is an isang protozoan. Meron silang incystation and excistation, the transformation from one morphology to another. If the motility or if the motility of Giardia lambia trophocyte is being diminished by the gastric juices, which will reduce the infection, then Giardia lambia has to convert, has to transform morphologically from being a trophozoite to cyst. Okay? Peristalsis, motion of cilia, human reflexa serves to expel parasite. Okay? That is what we call the physiologic function of our body. These physiologic functions of our body enables us to inhibit parasitic infection. Eh bakit? Yung cilia, minumove niya yung egg, minumove niya yung cyst, so that this cyst will be excreted through stool via defecation, so that the incubation period will not take place in the human body. You are exposed, but you were able to eliminate it, then that's a good sign of our host defense mechanism. On the other hand, coughing enables the expectoration of Ascaris lumbricoides and Paragonimus zostermani. Adult form of Ascaris lumbricoides can be expectorated through coughing. Umubo ka, sumuka ka. The adult worm, which can invade that area, can be eliminated, can be expectorated. Malalabas mo siya. Kung siya ay mayroong male and female inside the human body, then there will be a multiplication. But you were able to eliminate the female. Then even though that there is a male ascaris inside the body, pwede silang ma-eliminate. Urination also decreases Trichomonas vaginalis. Okay? You might wonder, paano yun? Magkaiba naman po yung urethra and the vaginal canal. If the Trichomonas vaginalis is within the vaginal area or the labia, then it can be flushed through urination. Okay? Remember that uh, before I proceed to this one, remember that in the event that the parasite is able to overcome physical barriers, no, the second host defense mechanism comes into play. Meaning to say, these are all our first line of defenses. If they are able to survive into our first line of defense, then the second line of defense has to come. Okay? The penetration of body's barrier resulted in a series of events that facilitate sensing of the invading parasites. Now, this is via what we call pathogen-associated molecular patterns. Okay? This is a response that enables the body to mount an immune response to act to eliminate these foreign bodies. If not totally eliminate, to limit the infection that is caused by these foreign bodies. Now, how about the host immune response? Take note that the host possesses both innate and acquired immunity, both of which defenses rely primarily on the humoral and cell-mediated mechanism of action. Take note that innate response happens when the body detects and eliminates pathogens. Okay? Uh, through what? Through non-specific mechanism. Okay? It can use mechanical, chemical, or cytokine-mediated methods to destroy or disrupt the invading organism or delay the time of invasion. One of the best method that the parasite or that the body can do is what we call the phagocytosis. Phagocytosis is divided into four phases: initiation, seek, uh, completion, digestion. No, yung eyes, if you still remember that, elimination and digestion. That's the mechanism of the body to eliminate. So that's the second line of defense through chemical or cytokine mediated method. Okay, through phagocytosis, macrophages and our dendritic cells subsequent pathogen elimination through what we call oxidation killing or oxidative killing. Okay, and they uses toxic agents or toxic peptides. Okay, parasite antigens can, this is can, hindi can stimulate the host to mount specific response to the antigen. Okay, now the parasite antigen originates from the surface, from the secretion and excretion, and from somatic tissues of the parasite. How about acquired immune response? What do we mean by acquired immune response? Acquired, from the name itself, acquired, nakuha mo siya. Okay? Remember guys that the immunity or the immune response to parasitic infections is under well-defined in genetic control and has a strong influence over the outcomes of the infection. Take note, the MHC. MHC stands for major histocompatibility complex. Okay, the MHC gene products help to regulate the T lymphocyte activities. Now, what will happen? Specific immune response now will begin to uh, uh, react with the parasitic antigens, and they are associated in what we call CD4 T cells or CD4 T helper lymphocytes. Okay, what is with that? Why is it important? Remember that T1 lymphocytes produce gamma interferon and interleukin 2. Okay, T1 lymphocytes produces gamma interferon and interleukin 2. Now, what about it? No? Ano ngayon? If it is capable of producing gamma interferon and interleukin 2. Remember, no, that these will activate the cytotoxic lymphocytes with CD8 surface marker that brings out the cell-mediated response. Okay? Remember that cell-mediated response has been observed in many parasitic infections. Take note of this. The cell-mediated immune response, no, has been observed, has been observed in most of the parasitic infections. Example of which is Toxocara canis. Toxocara canis is killed through cell-mediated activity. No? The migration of Toxocara canis. Ano ibig sabihin, sir, ng migration? Toxocara canis, no, meron siyang tinatawag na, what's the right term? Migrating larva. No? The migrating larva of Toxocara canis can be killed through the cell-mediated activity. On the other hand, no, kung si T1 nagpo-produce siya ng gamma interferon at saka ng interleukin 2 to activate cell-mediated immunity, T2 lymphocytes produces interleukin 4, interleukin 5, and interleukin 6, which is, no, in response for the immunoglobulin production. Take note that immunoglobulins or antibodies binds with specific parasite antigens and can activate complement. Now, once that the complement is being activated, then our immune response is okay. Take note, mostly, no, yung tataas or yung mataas na immunoglobulins or antibodies is our IgE, immunoglobulin E. No? Take note that in helminthic infections, eosinophilia and IgE level is increased. Eosinophilia means increase in eosinophil counts. Increase in eosinophils. Right? That is eosinophilia. And on the other hand, aside from eosinophilia, mataas din ang ating IgE. No? Eosinophilia is increased. Okay? Eosinophilia is increased and IgE is also increased. That is true with helminthic infections. Okay? Now, I want to make it clear. It doesn't mean na mayroon kang parasitic infection, you have eosinophilias or eosinophilia, because this is the most common misconception of the students. Kapag sinabing eosinophilia, oh, you have parasitic infections. Yes, of course. Parasitic infections will cause an increase in eosinophil count. But it doesn't mean all parasitic infection can do that. Eosinophilia is more common in helminthic infections. Other No, parasites such as protozoans, seldom or sometimes rare. Possible but rare. Unlike in helminthic infections, eosinophilia is common. Most especially if the helminthic infection or the helminthic parasite is a tissue invader. Okay? So this is what I'm telling a while ago. Visceral larva migrants. No? The visceral larval infection or the visceral larval migrants. VLM ang tawag natin dyan. Okay? We call it VLM. Visceral larval migrants. So sino yun? Si Parastrongylus cantonensis and we have your Toxocara. Okay? Ig antibodies mediate expulsion of the adult helminth in the GIT. Okay? And IgE identified on inflammatory cells. Now, we also have here what we call, okay, you might wonder, bakit nga ba nataas ang IgE? Okay? IgE antibodies are bound to the mucosal mast cells, eosinophils, and goblet cells that can mediate eventual expulsion of the adult, no, adult, so yun, adult GI, helminths in the GIT. So kaya siya nataas to expel this uh, helminths in the gastrointestinal tract. Right. Okay. Next in line is we have the type 1 hypersensitivity reaction. Type 1 hypersensitivity reaction, on the other hand, is common into what we call infection of Echinococcus granulosus 2, hydatid cyst. The same immediate hypersensitivity reaction has been observed in the site of the several arthropods. Right. Okay. Now, remember that the combined activity of IgG and IgM ano yan, prevents the penetration of uh, let me clarify this. The combination of the activity of IgG and IgM, let me clarify this statement, okay? The combined activity of IgG and IgM can prevent the penetration of erythrocytes by Plasmodium. Okay? Because remember, Plasmodium and Babesia are blood parasites. They invade the RBC. Meaning to say, the activity of IgM and IgG, the combined activity of these two immunoglobulins, of these two antibodies, will prohibit the invasion, will prohibit the penetration of Plasmodium and Babesia to the red cells, which can cause destruction to the red cells if the Plasmodium were able to invade erythrocytes. Okay? However, the combined activity of IgG and IgM is not effective against gastrointestinal helminths. What is effective for gastrointestinal helminths? We have our IgE. Okay? Now, how about the parasite invasion mechanism? Take note, people, that parasites have several characteristics that make it difficult for the host to detect and eliminate them. The parasite size, complicated life cycles, location within the body are relatively the reasons why no the parasites cannot be detected easily. In addition to that, there are adaptations of the parasites. Okay, adaptations that develop the mechanism of various parasites. Now we have here what we call resistance to immune response, immune suppression, no, antigenic variation, host mimicry, and intracellular sequestration. These are the parasite's mechanism of invasion. So let's discuss one by one. When we said resistance to immune response, take note, people, that protozoans and helminthic infections that enter via the bloodstream or tissue are often able to survive and replicate. Why? Because they are resistant to the host's innate immune response. Okay? They are immune. Macrophages can phagocytose protozoa, but the cuticle of the helminths makes them resistant to the macrophage. Therefore, no, effects on both neutrophils and macrophage is not effective. Now, why is it helminths can resist the macrophage aside from the cuticle and the integument? This is due to the loss of the surface molecule that binds the complement or the acquisition of the host regulatory proteins, such as our uh, accelerating factors. Okay, we also have your apolipoprotein L, which destroys the trypanosoma, no? With exception to trypanosoma drusi. Now, how about immune suppression? Remember, guys, that there are parasites that can reduce the immune function of the macrophage. Now, what will happen? If the parasites can able or are capable of reducing the function of the macrophage, it resulted to the lower capacity of phagocytosis and effective processing of antigen. Now, in this case, we have what we call Plasmodium species. No? Plasmodium species has the capability to lower the capacity of phagocytosis. Kaya nga sila nakakapag-invade ng RBC. Entamoeba histolytica, on the other hand, suppresses the macrophage respiratory burst. No? And can consequently end the consequent nitric oxide production, which produces suppressor factor that can inhibit the movement of the monocytes to the site of invasion. Okay? Remember also that in infection of Fasciola hepatica, there is a downregulation of T1 lymphocytes. On the other hand, for filariasis, Pochereria bancrofti, and Brugia malayi, no, there is a polyclonal hypergammaglobulinemia. Ano yung meron doon? Where antibodies lack specificity. Therefore, the parasite can survive. Lack of specificity. Remember, that's one of the requirements. Kailangan specific so that there will be a reaction. And remember that the blocking, no, before I proceed to this, remember that the blocking of antibodies produced by the several parasites, like what I've mentioned a while ago, Wuchereria bancrofti, can also dampen the effect of immune response. For Necator americanus infection, no, the immune response is directed against the deeper layers of its cuticle. Now, how about for Cysticercus celluaceae? Cysticercus celluaceae is the infective stage of Tania. Na? Tania. Okay? So what is with this one? Uh, Cysticercus celluaceae's infection suppress inflammatory response. And take note Plasmodium species and Trypanosoma cruzi lead to immunosuppression through immune complexes. And lastly, Schistosoma species complement cannot participate. Complement is consumed by soluble antigens. Third is we have antigenic variation. Example of which is Trypanosoma brucei. In Trypanosoma brucei infection, the initial host response against the surface glycoproteins of the trypomastocyte is very effective. But, but in the subsequent releases of the trypomastocyte, the immune response is no longer effective since the parasite has changed already the changed already the antigenic profile. Kasi they were able to detect, oh, the human body is capable to suppress the trypomastocyte being released. That we have to change the genetic variation, change the genetic composition, antigenic composition. Then the immune response of the human against that antigenic variation is not anymore. It's not anymore uh, applicable. The surface protein variation has also been observed in Georgia lambia. Now, how about for malarial species? Malarial parasites, especially Plasmodium falciparum, exhibit antigenic diversity. This is a mechanism through repeat variation of encoded polypeptides. No? And these repeat sequences are antigenic epitopes. Kaya si Plasmodium falciparum siya yung deadliest eh. Of all Plasmodium strains, of all Plasmodium species, si Plasmodium falciparum yung dangerous. Most deadliest, no? Most yung most dangerous. Siya din yung deadliest. Bakit? Capable siya mag-rupture of RBC in a single period of time that causes chills and fever to the patient. We also have host mimicry. Mimicry, ginagaya. No? The larval stage of Echinococcus granulosus carry P blood group antigen. No? Remember also that Echinococcus granulosus larval stage has been found to carry that group, and the tegument of the schistosoma species acquire antigenic molecule from the host. Sina-acquire niya yung response ni host para hindi siya ma-eliminate so that this parasite can cause infection. Antibodies produced against the parasite then fail to recognize non-self from self-antigen. Anong ginagawa dito? Instead of killing the parasite, nare-recognize pa niya ang napapatay pa niya, yung self-antigen niya. Okay? Now, lastly, we have the intracellular sequestration. Amastigotes of Trypanosoma and Leishmania, no? Trypanosoma cruzi to be exact, and Leishmania species proliferate in the macrophages in various organs. Now, for Toxoplasma gondii, it multiplied inside the macrophage as well as in the nucleated cells. Now, the late intracellular stages of falciparum, Plasmodium falciparum, are sequestered from the circulation in deep vasculature beds. Okay? Take note, people, that this sequestration process exclude the parasitized red cells from the splenic filtration and the action of the antibodies. Okay? Now, for the groups of medically important parasites, we have their protozoa. No? When we said protozoans, they are unicellular. No? Parasitic infections are either due to unicellular protozoans or multicellular metazoans. Take note, guys, that generally protozoans parasites are provided with nucleus and nuclei as well as cytoplasm. And of course, the outer covering or outer laminating membrane. They have organelles, locomotoric apparatus, or for their locomotion, the cilia, flagella, and pseudopods. And take note that all protozoans, no, fall under the kingdom Protista, which is a diverse group of eukaryotic organisms. We have here the classifications of protozoans. We have the uh, Sarcomastigophora. No? Under Sarcomastigophora, we have subphylum Sarcordina, no? Class Lobicea, and we have the amoebas. Under phylum Sarcomastigophora, we have phylum Mastigophora. We have there your class Zoomastigophora, and we have there your flagellates, no? And we have your hemoflagellates. Under phylum Siliphora, we have Kinetophragminophorea. We have there the ciliates. And for Apicomplexa, we have there the Babesia, class Porozoa, and we have there Plasmodium as well as our Babesia. Some of our miscellaneous protozoans, such as Cyclospora cayotinensis, Cytoisospora belli, are classified under Apicocomplex. Now, for subkingdom Metazoa, no, we have naman the helminths, no? The phylum Nemahelminthes and phylum Platyhelminthes. Basically, Nemahelminthes are rounded except for Filaria. Now, so we have here the class Nematoda, which are the roundworms, Ascaris, Enterobius, and so on and so forth. For class Filariae, we have the Filaria, Wuchereria bancrofti, Rugia malayi. They are flat, but they are classified under phylum Nemahelminthes. For phylum Platyhelminthes, we have there the flatworms or the tapeworms and the flukes. Tapeworms, ito yung mga cestodes, and fluke is our uh, trematodes. For kingdom Animalia, we have there the phylum Arthropoda, arthropods. Now we have the Crustacea, Arachnida, no, Insecta, Siliphora, and Pentastomida. And that is the end of our lecture number 1, part